Thanks very much, Anna. I just want to echo the sentiments of the previous speakers and passing on my thanks to the course organisers for giving me this opportunity to speak today. So, in clinical practice, it is clear that there is notable variation between patients in response to a given drug, affecting both efficacy and toxicity. This interindividual variation is important when you consider the wider context that global expenditure on drugs is forecast to reach $1.4 trillion by next year to pay for 4.5 trillion doses. Yet it's estimated that only 1/2 to maximally 3/4 of patients respond beneficially to first-line pharmacotherapy across a broad range of disease areas. 6 to 7% of hospitalizations are attributable to an adverse drug reaction, and 15% of patients experience an adverse drug reaction. And therefore, it's paramount that we study, understand, and act on the factors which are involved in this heterogeneous drug response that we see in order to improve the safety, efficacy, and cost-effectiveness of our prescription pharmacotherapy. There are multiple different factors which are involved in variable drug response, including demographics, comorbidities, environmental exposures, drug-drug and drug-food interactions, and genomic variation, which we're focusing on here today. Pharmacogenomics is a study of the genetic determinants of drug response. And there's been an area of intense research, and I totted up last night that now there are over 200 drugs which contain genomic information somewhere within their FDA-approved label. Every time I have a look, that number's increased. And here are some examples of some of the more recognized and stronger drug-gene associations. Yet it's clear that the majority of these genomic variants are common. And secondly, as this list of drug-gene associations grows, it's going to become harder and harder to conduct more traditional prospective randomized studies to determine the effectiveness. However, there's been an evolution of pharmacogenomics as we've moved from the candidate gene era through GWAS now into the next-gen sequencing area. And that's been coupled with these huge big data endeavors, such as the 100,000 Genomes Project. We've heard about the All of Us initiative in America. China has embarked on a project. And in fact, actually, there's now over 10 countries involved in carrying out whole genome sequencing of 100,000 of their population. And so this gives us an unparalleled opportunity now to begin to hone in and understand what might be the impact of rare variants on drug response. Yet before we do that, I wondered if it was worth looking back to think, are there any clues that rare variation might be important in drug response? And I'd advocate that there are several. One example is with malignant hyperthermia. Fevers due to an anesthetic were documented at the start of the 12th century. The first cases were in 1900, and then in 1962, around the same time that the term pharmacogenetics was coined, Dr. Denver in Melbourne described 11 malignant hyperthermia reactions in a family and realized that this was a genetic condition with an autosomal dominant pattern of inheritance, and it's severe. Patients who are undergoing an anesthetic with one of these volatile halogenated agents or succinylcholine can experience hypermetabolism, tachycardia, dropping blood pressure, hyperthermia, and it's fatal. It kills. It's rare, but it is fatal. Fortunately, the introduction of dantrolene and muscle relaxants has helped to reduce that fatality rate. And importantly, it's been associated with now 50 different rare genetic variants, clustering in the ryanodine receptor 1 gene, but also in the alpha subunit of the dihydropyridine receptor. And through aberrant pharmacodynamic pathways, these seem to be involved in leading to excess release of intracellular calcium into the skeletal cells, leading to excess muscle contraction and the subsequent deleterious clinical sequelae. I'm fortunate, as I said, I'm a Health Education England Fellow, and I'm involved in a study using the data now from the 100,000 Genomes Project to, first of all, characterize variation within pharmacogenes, and then we'll be moving on as the drug data becomes available to conduct case-control studies to test the hypothesis that rare variants in some of these associated genes have the same impact as the common variant within those genes. And finally, we'll do some cost-consequence analyses. So this now represents really a first dive for me into the research environment, and I've been analysing successfully extracted data on 195 genes, both PK, and I wanted to also focus on some of the pharmacodynamic genes from just over 60,000 participants, and extracted 2 million variants. 90% are single nucleotide variants and 10% were indels. And we've seen this before, but it really struck me that 94% of all these different variants have a minor allele frequency of less than 0.1%. Actually, all of the common variants that we associate with drug response, they sit in just that 2.8% at the top of all the different variants present in our genome. And when you stratify by type of variant, it seems that indels are a little bit more common, but still 90% of them are present in less than 0.1% of the population. Then I wanted to have a look at the different variant classes within this 191 genes. 95% sit in the modifiers group, which includes mostly intron variants, but there'll also be some in the regulatory regions. 3% are in what the ensemble sequence ontology classifies a high, moderate, or low impact variant. Moderate is mainly driven by missense variants, low by synonymous variants, and high by a range of splice frameshifts and variants and the like. When you then open up that, you see that it's about 58% of all the variants are missense, 28 are synonymous, but it's actually almost 10% are splice variants, either splice acceptor or splice donor variants, and then there's smaller proportions of the other classes of variants. And then when you look at this on a gene level, you see that that is reflected across the genes. These graphs aren't actually yet standardized for the length of the gene, but they illustrate that in these phase 1 drug metabolizing enzymes, there's a lot of variation, in particular, a relatively high amount of high impact variants in red there. And that's reflected again in the phase 2 enzymes and in the transporters. But when you begin to look at some of the genes involved in pharmacodynamics, particularly these in oncology, and also drugs which I labeled as neuropsychiatric drugs, so they involve genes encoding for the reuptake transporters of catecholamines, serotonin, and dopamine. Actually, the prevalence of the high-impact variants was much reduced, and similarly with the nuclear receptors, it was much lower, suggesting that these genes are under more stringent evolutionary pressures to maintain function. The next thing I did was I took out the high and the moderate impact variants, so-called putatively functional, and then used the ensemble variant effect predictor in order to try and determine for the missense variants whether they're potentially deleterious or not. And at the moment I use SIFT and polyphen algorithms. There are other more advanced and pharmacologically relevant algorithms I want to use as well. And I wanted to then see, out of all of the potential functional variation in each of these genes, how much is attributable to common or rare variants. And in the CYP enzymes, there's an awful lot which seems to be common variation. Maybe that's why we know more about them at the moment. But ultimately, the proportion really varies on a gene-by-gene basis. And as you move through the phase 2 and the transporters, more and more seem to have the majority of their variation in these coding regions is attributable to these rare variants. And that follows. And so overall, when I totted it up, of these 195 genes, actually over half of them, all of this putative functional variation appears to be rare. And then what I wanted to ask, well, if a patient sits down in front of me, how many functional variants may that person in front of me have on average in these 195 pharmacogenes? And the answer is about 37. And then how many of those are rare? Well, it's about 11%, so around about 4 variants per participant. That 11% I feel is not insignificant, given that these rare variants are often enriched for deleterious consequences. Clearly, this is not the only endeavour to have undertaken such work, and moving forward, I want to ensure it is novel by, for example, using some of the key attributes of the 100,000 Genomes Project. So using the trio structure from the rare disease arm, be able to assess the rates of de novo variation, and using the cancer arm, be able to assess the degree of variation within these genes within the somatic genome. And as Ellie said, this is it's a collaborative, it's a huge endeavour. And so if other people are interested, please get in contact. I just now wanted to, the final bit, just touch on something a little bit different, and this takes me right back to how I work clinically, where it does really seem that there are multiple different factors which are attributable to drug response. And I've worked with a cardiovascular cohort which recruited across England over 1,400 patients hospitalized with an acute coronary syndrome, a heart attack, and followed them up for at least a year. And I was interested in seeing, trying to find out variants that affect some of the drugs that they're on. In particular, we looked at Atorvastatin, bisoprolol, and clopidogrel. And this just highlights the Atorvastatin, some of its main major metabolites, both the hydroxy species and the lactone species. And with a colleague here, Vanessa, we developed and validated an assay according to European Medicines Agency guidelines, and then we applied it to over 1,000 patients within this cohort. And then the first layer I wanted to ask was, what clinical factors are associated with variable exposure to atorvastatin or its metabolites or those metabolic ratios? And you see some of the usual suspects here, age, sex, BMI, but also some potentially novel drug-drug interactions, which was interesting. And then I wanted to adjust for that and then go on to do genome-wide association studies. And I'm afraid this is not yet published, it's getting there, but this shows, with one of the metabolic ratios, a very clear signal, and the p-value is about 10 to the minus 16. It's not at the minus 221 of Professor Roden's, but I still think it's not bad. And then on top of this, I then thought, well, I wonder if I can do anything with rare variation. The sample size is 560. For disease genetics, that would be too little, but we know, as already mentioned, that some of the odds ratios for pharmacogenetic associations are much stronger than they are for disease associations. So I thought, I wonder if I can find something with rare variation. And we'd fortunately genotyped these patients on the Illumina Omni Express exome chip. So they weren't sequenced, but it was a chip enriched for rare variants. And so this was my first go with a colleague, Eunice. It was done with these patients, and each dot represents a gene unit, so not an individual variant, but the genes, variants within that gene which we had typed, which were predicted to be deleterious in at least one of 5 different computer algorithms, were grouped together. And the red line shows the statistical significance threshold after adjusting for multiple testing. And although nothing reaches that, there's an interesting association in chromosome 8, the top association, which actually is in a novel drug transporter. So there's some potential biological plausibility there. And it had a collective minor allele frequency of just under 2%. We now need to go on and do biological studies in order to try and understand this signal further. But when you put it all together, so how much of this metabolic ratio can I explain? It's about 30%. 16% is due to those clinical factors. There's another 11% due to those common genetic factors. And this rare variant adds on another 1%. And that 1% doesn't perhaps sound like much, but for those individual patients carrying it, actually, a quarter of those made up the top 3% of this, the top end of this metabolic ratio. And I do feel moving forward, we're going to need to be identifying and putting this all together and using the big data available to us in order to try and better pass interindividual drug response variability to help our patients. So thank you very much for listening. Thank you to Munir, the supervisor, and everyone else at Liverpool. And also, yeah, thanks to Health Education England for my fellowship and the 100,000 Genomes Project for making the data available so that I'm able to dive in and wear my bioinformatics hat. Thank you very much.